Okay, so we resume our lecture for today, and today's lecture will be the last lecture, okay, since we only have 6 LUs. And I hope that you guys go through what I've posted on Telegram and also Elite for LU5. Okay. So it was the bite-sized session. Right, so today's lecture is on toxic waste and site cleanup. Yeah. So that's the title of the LU. And we are going to look at toxic waste treatments, the 3 types of non-biotech treatments, chemical, physical, and thermal. And we go to the biotech method in the 3rd subtopic, which is bioremediation. Okay, so basically we are going to look at how we want to tackle toxic waste treatments using biotech methods, but also we want to have a look at how those non-biotech methods can be done in order to tackle the toxic waste. Is it everything clear? Before that, is it clear, the slide, my voice?
Yes, doctor.
Okay, thanks. Right. Okay, thanks. Okay, first, let's have a look at the introduction. Yeah. So when we talk about toxic materials, what are toxic materials? Okay, thanks. Toxic materials are poisonous byproducts. That is defined by poisonous byproducts, the products that give, that are poisonous, that give harm. Yeah? And where do these toxic materials come from? Actually, it can be resulted naturally, not only from man-made processes. Here, I just mentioned about the result of industries, but it can be also resulted from natural processes. But since the evolution of industry is increasing, yeah, so there are also a large amount of toxic materials that come from the man-made processes, yeah? Normally from the industry, from whatever the operations of the plants, yeah. For example, the manufacturing industry, the farming, construction, automotive, laboratories, hospitals, yeah. So all these activities, they emit toxic materials, yeah. And those toxic materials, it can be heavy metals, radiation, yeah? Dangerous pathogens. Pathogens are those microbes that can cause diseases, yeah? Or harms to the human and environment. And other type of toxics. Toxic materials are just a generic term, but it can be many forms of dangerous compounds. It has become more abundant since the Industrial Revolution, causing serious global health issues. Disposing of such waste has become even more critical with the addition of numerous technological advances containing toxic chemical components. For example, the use of the creation of chemicals like from, like the toiletries, yeah. So those are some, where the toxic compounds come from, the detergents, the chemicals used in the laboratories, yeah. There's so many actually. And one of them is also the electronics, the products from the electronics, the items, electronics, such as cellular phones, computers, televisions, and also solar panels. They also emit toxic chemicals that can harm the environment if they are not disposed properly. Yeah. All right. So a material is considered toxic when it causes death or harm by being inhaled, swallowed, or absorbed through the skin. Okay. Right. So one of those sources of toxic materials are, as I mentioned just now, are electronic items. I think this one should be covered a little bit in recycling, but anyway, I just covered here. Okay. So electronic items are one of the accumulating products, right? in every household. Yeah? So it is said from statistics that the average American household has more than 10 electronics. Yeah? At least 2 of which are cell phones. I don't have the statistics for Malaysia. I'm sorry for that. This one I just got from literature. Yeah, but it shows that actually it's not just in America, but also in other countries, including Malaysia. We are accumulating electronic items, right? So once the electronic items, perhaps they are not like functioning, they are not used anymore. So what happened to those things? Currently, we might not have that really much places that can recycle electronic items, but actually they are actually such practice. Such practice, maybe I'm not sure in Kuching, but maybe in KL, maybe there are some areas, but it's not that really wide lah, the practice. Yeah, but the right way to actually dispose or tackle the problem of e-waste, it's called e-waste and electronic waste, is to recycle them, right? Instead of disposing those waste to the landfill, because landfill is normally the last option, the last result. And it's not that really a favorable option, but at the moment we don't have much choice because most of the waste just go to the landfill. Yeah? But actually it's not the ideal option for disposing the electronics. The reason is because electronic items, they are not supposed to be at the landfill. I mean, there are things that can be actually recycled from those items. For example, all these valuable materials like copper, tin, iron, aluminium, fossil fuels, titanium, gold, and silver. Those things can actually be obtained from the electronic items, and they can be reused, recycled to make new items, new materials. Okay, so the ideal way of tackling the e-waste is actually to recover, reuse, and recycle the electronic waste. Yeah. So there is a statement that says recycling 1 million cell phones can recover 75 pounds of gold, 772 pounds of silver, and 35,000 pounds of copper, and 33 pounds of palladium. So that is the finding from recycling the electronic waste. Is from 1 million cell phones can produce all this amount of recyclables. And there is also a statement says that Apple revealed that it recovered 2,204 pounds of gold worth $40 million from recycled iPhones, Macs, and iPads in 2015. So that is actually the benefit of recycling. Instead of these things accumulated at the landfill, and what happened throughout the time, perhaps all those the components might leak to the ground and cause pollution to the ground. Yeah, because those things are actually toxic. Yeah, so by nature. So those are the options lah. Instead of disposing these toxic waste, we can actually do something on that by recycling or reusing them. Okay, right. So let's go back to the toxic materials. Right. So these are some of the key substances listed by United Nations Environment Programme, whereby these compounds are those that can pose a risk to human health. So, for example, arsenic, asbestos, cadmium, chromium, clinical waste from the health industry, from hospitals, laboratories, cyanide, poison in pesticides, lead, batteries found in batteries, paints, mercury, polychlorinated biphenyls, PCBs, persistent organic pollutants, POPs, and also acid and alkalis. So those are examples of toxic compounds, and they can be obtained from all of these sources. And actually, it's not just, as I said just now, it's not just from the man-made processes or items, but arsenic, asbestos, they are also present naturally. But perhaps when we talk about natural occurrence of these compounds, there might be just certain places. It's not that really wide out there in the environment. Yeah? With the increasing amount of the with the industrial products, actually, the industrial products, the amount is overriding the natural amount of these toxic compounds. But you have to know also these compounds also present naturally. Okay, so these are the toxic materials. And as biotechnologists, why we study this course, we study the environmental science, and also we study the biotech approaches, right? So that makes the difference between this course and environmental science course. And when we talk about environmental science, we're just talking about what happened to the environment, what are the, you know, the toxic materials, but they might not discuss from biotech point of view. So that's the difference. So as biotechnologists, you guys must have that sort of how to say that, sort of perspective, how you want to treat, how you want to tackle this kind of toxic waste, toxic materials from biotech point of view. Yeah, because biotech by nature is a field that tries to solve the problems, right? And one of them is these environmental problems, the pollution and the contamination. Okay, so here I include some of the research that have been done, yeah, by biotechnologists all around the world. Yeah. So these are all the reports, some of the reports actually. So you can see that I just highlight the headline over here. So arsenic toxicity, bioremediation, it can be actually tackled by using bacteria, yeah? Indigenous bacteria isolated from middle Gangetic plain of Bihar, India. So, I mean, these researchers, they isolate the river, the samples from the river, and they found out that there are bacteria that can degrade arsenic. Yeah, so we know that just now arsenic is a toxic compound, isn't it? And then you can see another one here, another research done on bioremediation of cadmium. Yeah? Contaminated soil using symbiosis between leguminous plant and recombinant rhizobia with the MTL4 and the PCS genes. Okay, so here we can see that it's not just microorganisms. We can also utilize plant. Yeah? This one is like symbiosis. It's a interaction between the plant and also the bacteria. Yeah, in order to treat to reduce the cadmium. So the objective of bioremediation is to reduce these toxic compounds into byproducts that are not harmful, so that these toxic compounds, the arsenic, the cadmium, are no longer in the environment. And that is the objective of bioremediation. And how they are reduced is by the action of the biological organism, yeah? Be it microorganisms or plants. Yeah? All right, and then you see the third article here, asbestos pollution using fungi, yeah, using fungi, right? And then bioremediation of chromium solutions and chromium containing wastewater. Yeah, you can see there are actually the research into using the microorganisms is actually there are a lot, and it's not just one type of bacteria. Like for example, let's say chromium. There are not only one type of bacteria that can degrade it, but there are actually many types of bacteria. And perhaps let's say this group of researchers, they just found this particular group of bacteria. But actually, if let's say you do more research, you can find like maybe other types of other groups of bacteria isolated from other places. They can also do the same thing. Yeah. So that's research. That's, I mean, there is no end point of research. Yeah, so if you ask me like how many types of bacteria that can degrade cadmium, I don't have the answer because the number of researchers is increasing from time to time, and the number of findings, the results obtained is increasing as well. Yeah, so there is no endpoint for the research, including in biotech, in solving this kind of problems. Okay. All right, another research here, biodegradation of cyanide waste from mining and jewelry industries. So this cyanide is produced from examples of the sources are from the mining and jewelry industries. So this group of researchers, they study about how to degrade the cyanide using the bacterium. You can see from the articles here they are using. They found this bacteria that is capable to degrade the cyanide, Pseudomonas pseudoalkaligenes. Yeah? So some of these microbes, they are actually obtained naturally, or what we call wild type, or you can also you will also notice the word indigenous. Indigenous means indigenous means like pure, right? Something that is unaltered, something that is natural, right? So some of the microorganisms, they are obtained naturally, whenever like the researchers screen for certain microbes, they found out that the microbes that are obtained naturally, they can do the bioremediation, right? But some of the microbes, they are also recombinant. Yeah? So recombinant. So, but the recombinant microbes, they are actually derived from the understanding about the indigenous microbes. Okay, perhaps let's say the researchers, they found out that certain type of wild type microbes can degrade, for example, cyanide. Okay, and then but the problems with the wild type normally is that their performance might not be that really might be low in certain conditions, but so how to improve that performance? So that comes genetic engineering or recombinant DNA technology, but it's actually based on the information or understanding about the wild type mechanism. Okay, so when people found out that these are the wild type, they have this gene that can degrade cyanide, yeah, for example, or other types of organic toxic materials. So they use that gene and then clone it into new hosts, new organism that can perhaps produce more proteins or more enzymes that can degrade that particular compound, right? So it means that the performance of the recombinant microorganism in degrading the toxic materials will be better than the natural or the wild type organisms. Okay, so that's the difference. But it can happen in both ways, using indigenous or wild type organisms or recombinant microorganisms. Okay, right. So if you see here the name of the microbes, if I just go back to the previous one, so where is the name of the microbes? So the name of the microbes, you might notice that it's not that really common because these microbes, how they found out the microbes, let's say, for example, the arsenic degrading microbes. Where do they found these microbes? They found this microbe from the places that have that particular compounds, that arsenic. Let's say the naturally occurring microbes that can degrade arsenic. They are actually obtained from the places that have the arsenic. The reason is because why those microbes are there is because like the microbes will go for something that they can eat, right? That is the nature of organisms, right? So if like if let's say you have like food, you put food outside your house. So after some time, there will be cats or dogs that will approach the food, right? So that is the nature of organism. So the organism will go towards the source of food. And the same thing goes for microorganisms. So how you want to find out those microbes that can do whatever the things, do certain kind of degradation, you look for places that have that sort of compounds. Yeah, as I said just now, these some of the toxic compounds, they are also present naturally. Yeah? So those places actually, they have that microbes that can degrade the compounds. Okay. Um, yeah, okay. So yeah, let's have a look at some of other articles. So this one is lead absorption mechanisms in bacteria as strategies for lead bioremediation. Yeah. So you can see that actually every type of toxic waste, they are microorganisms that can degrade those compounds. So that's lead, and then this one is mercury. Yeah, mercury. So this one is, what's the type of, what's the name of it? It doesn't have the name. Yeah. But another term that you can see here, if you see here, mercury resistant bacteria. So the microbes that, whatever the toxic compounds are, they are also called like, for example, just now, lead degrading bacteria, for example. This is just to introduce you the terms so that you know what those terms mean. And sometimes they are also used like lead resistant bacteria. This is just to emphasize the terminologies. So this one you see here. So lead degrading bacteria, you know that it means that the bacteria can degrade the lead. Yeah? But this term is also used, like lead resistant bacteria is also used, because the reason why, because the bacteria, they are resistant towards lead. They are resistant. They can survive with the lead. They actually eat this kind of waste. So that's why they are called lead resistant bacteria. But actually they refer to the same thing. Yeah, just for you to know that, so that you don't confuse when you read the articles having those terms. So it can be lead-resistant bacteria, mercury-resistant bacteria, what else? The cyanide-resistant bacteria or fungi. Yeah? So the word resistant degrading is actually gives you the same meaning that the microbes can survive with the existence of those things. All right, this one is polyfluorinated biphenyls, POBs. Yeah, POBs. And then this one is persistent organic pollutants. You can just actually type whatever the name of the toxic materials and find the articles related to it, the bioremediation of those things. Okay, so you can see the name of the species over here, 2 bacterial species. Yeah, you can see that the name is not that really common because those bacteria are quite rare. They are not like exist in a common environment. Okay. Right. So those are actually the biotech approaches of how to tackle the toxic waste accumulation, yeah, in the environment. Yeah. So like we are biotechnology, of course, we are concerned with the biotech methods, but actually these toxic compounds, they can also solved by using chemical treatment. Yeah, chemical treatment. What else? Physical treatment, and also thermal treatment. Yeah? So these are other options, yeah, other than biotech. But what is the disadvantages of these methods is that they are not environmentally friendly, one thing, and then complex, is difficult, and expensive. And they might also emit hazardous byproducts from the degradation process. Yeah? So that's the disadvantages of non-biotech methods. Okay, so biotech methods, we know that biotech methods are more environmentally friendly because they are natural processes. Yeah? So it does actually emit side products, but perhaps the side products might not that as harmful as those side products produced by the chemical processes. Okay. Right. Okay, so toxic waste treatment, we are going to look at the three non-biotech methods, yeah, chemical, physical, and thermal treatment. Okay, so by right, you should know how to define, how whatever the biotech approach method is defined. Okay, although we study about environmental biotech, but some of the things that we study in this course are not necessarily biotech based. So how to know whether they are biotech based or not? You have to understand the concept of biotech, isn't it? So biotech approaches, biotech methods are those methods that utilize biological organism. Yeah, that's the easiest way. Anything, it must involve the biotech, the biological organism. If you're just using like chemicals, that is not a biotech approach. Although we study like, like chemical treatment in this course. Okay. So I leave it to you for you to differentiate lah. You have to understand. You're already in final semester. So after this, when you attend any interview, when people ask you what is biotechnology, I hope that you can answer what is biotech, the principle of biotech, okay, that will lead you to understand everything from biotech's perspective, okay? All right, so let's have a look at the toxic waste treatments. We focus on the non-biotech methods first, and at the end, we'll look for the bioremediation. So as I said just now, the treatment of these toxic waste, whatever the toxic waste, it can be also treated by using chemical treatment, yeah? Other than biotech, chemical treatment. So what are the methods under chemical treatment? Chemical treatments is quite easy for you to visualize. It's something related to the chemical changes, or you use chemicals, you add chemicals to the processes, right? So that is the chemical processes. So the chemical treatments for the toxic waste, there are 5 here, yeah, that we're going to look at. The first one is solubility, neutralization, precipitation, color removal, and disinfection. I don't have the explanation for solubility. Solubility perhaps is something related to how those thing is, how the reaction of the chemicals. No, it's not stated right. Okay, let's have a look at the neutralization. Okay, neutralization. So some of the toxic materials mentioned just now are acid and alkali. Acidic wastewater or basic wastewater. So how to treat it? So is by using the chemical methods, acidic wastewater, the basic thing is to neutralize, because anything that is acidic might not be as favorable as something that is neutral. So the basic concept is to neutralize acidic or basic, whatever that is acidic or basic, right? So if let's say the wastewater is acidic, how to treat it is like by neutralizing it by adding alkali, such as this lime, snake lime, because lime is basic in nature. Soda, sodium hydroxide, or calcium carbonate. So that could neutralize the acidic wastewater. Right. Okay, so this is the process of neutralization, neutralizing the acidic wastewater at site. Yeah, so you just basically add the alkali into it, and the neutralization process takes place. And of course, in order to know, it's like by monitoring the pH of the reaction, the reaction mixture. Okay, that is neutralization. And then precipitation. Precipitation is another type of chemical treatment. So how this process goes is by varying the pH levels, because the nature of certain, like most of the chemicals, they can form precipitates. They can precipitate by altering the pH values of the solution of the compounds. So the precipitation, the basic principles is by varying the pH levels, and yeah, depending on the metal ion, yeah, the different compounds have different pH values that can cause it to precipitate. Yeah, so at their optimal pH values, at the end, what will be resulted will be the precipitate or insoluble salt. So that's how the things can be removed. Whatever the toxic the is can be removed from the suspension. Okay, right. So this is a picture that shows precipitation tank for removal water impurities by precipitation. Right. The third method is color removal. Yeah, color removal. So the color color substance is also a contaminant that is released by, for example, the textile industry. Yeah, the textile industry, they produce lots of water contaminants to the sea, to the river, right? So those colors, I mean, those compounds, those colored compounds, they are actually toxics. Yeah. So how they can be removed from the water bodies, if you are using the chemical treatment, yeah, so it can be treated using carbon adsorption, coagulation, flocculation, or chemical oxidation by adding certain types of chemicals like chlorine or other strong oxidizers. Okay, so carbon adsorption is by adding carbon actually. So this carbon will absorb the colors so that the colors will not be accumulated in the water bodies. Yeah? Right. So there are several types of chemicals that can actually react with the colors and then attract it and perhaps form coagulate. Yeah? It forms like certain like for example here you can see this is amount of the things that attracted by the addition of the chemicals. Yeah, so these colored compounds can be removed later on. Yeah? All right, and then disinfection. This is a very popular or common method, even in the wastewater treatment and also the water treatment, the clean water treatment, the one that comes out from your taps. So what the disinfection is, so this is by adding certain types of chemicals. The common types of chemical is chlorine. Yeah? The reason why chlorine is used is because it is effective at low concentration, and also it is relatively cheaper compared to other options like ozone. Yeah? So ozone is another option. But the disadvantages of chlorine is that it's a poisonous yellow-green gas. Yeah, although it is effective, but normally we don't have. I mean, for the water treatment, the people still opt for chlorine because of the effectiveness and also the cheap price. But the disadvantages of chlorine is that it is actually poisonous. Yeah, and also corrosive. It's corrosive. It can also cause some health problems like respiratory and eye irritation, or if let's say it's in extreme exposure. Another option of disinfectant is ozone, but the problem with ozone is that it is expensive, yeah? And also the process or the operation require expensive facilities. Yeah? So that's why ozone is not that very common as chlorine. Okay, right. So that's disinfection. So those are the chemical processes: disinfection, color removal, precipitation, and also neutralization. Okay. So those are how the toxic compounds can be removed from the water bodies or whatever the suspension. Eh? Right. Apart from the chemical treatment, the toxic materials can also be removed by using physical treatment. When we say physical treatment, is a processes that is conducted based on the physical features of whatever the suspension that contains the toxic compounds. So some of the methods under physical treatment, the first one is flotation. So this flotation, how the process is by sparging A. Like this is actually this is the suspension containing whatever the toxic compounds. The A is sparged into it, right? So as a result, the bubbles is formed, right? And these bubbles will attach to whatever the particles or droplets, or in this case is whatever the compounds that you want to remove. Yeah. So, and at the end, this the whole the mixture of the bubbles and the particles can be easily removed from the suspension. Okay. So the mechanism is by sparging the air in, form the bubbles, and the bubbles the bubbles that attach to the particles will be removed from the suspension. Okay, so these are examples of pictures that shows flotation at the industry. Yeah. This is at the newspaper, the paper industry. Yeah, flotation. Okay. The second method is sedimentation. Okay, this one you have come across this in the wastewater treatment. Yeah? During the primary treatment, where the wastewater is allowed to settle, right, in the settling tank. So what happens is that after some time, the suspension that contains different types of particles, it will sediment, yeah, according to the density. So that is the basic principles of the process. So based on the density, like those that are light, for example, the oil, it will be on the surface, right, of the suspension, whereas those that are heavy will be settled at the bottom. And the same thing for the toxic waste materials. Like this is just an example of the wastewater treatment. But if let's say in the case of suspension that contains the toxic materials, so once the suspension is allowed to sediment, but after some time, there would be different layers of compounds consisting of different materials, and that is how those materials can be removed. Yeah. Okay. The third way or method is centrifugation. So centrifugation, I know that you have come across the process in the lab, centrifuge. You use centrifuge, but that one is lab scale centrifuge. And centrifugation is actually quite a broad concept. It's not just for centrifuging whatever that you have in the lab, but also at the industry, it is used to separate suspension with different density, particles with different density. Okay? So that's why after you, if let's say you see the things in the lab, after you centrifuge, you will see pellets, right? Pellets and the liquid, which we call supernatant, right? So why those things are formed? Because those particles have different density. Yeah? Same as the sedimentation just now, but the difference between centrifugation and sedimentation is that sedimentation, you might allow the things to sediment naturally, right? I mean, like in the primary tank, the things settle by itself. But centrifugation, you apply centrifugal force. Centrifugal force. And that is the principle of it. The centrifugal force, whereby you can see that it is rotating, right, under certain speed, isn't it? So that is where the force is created, and that force trigger the separation of the particles according to the density. And at the end, you will see that the particles settle at different layers. Okay. So if you have like more than just supernatant and pellets, you might see like different things in between, So that is how different particles can be separated, can be removed. Yeah. from other particles. Okay, so these are the examples of large-scale centrifuge. Yeah, large-scale centrifuge. So if you study bioprocess last time, right, if you still remember, centrifugation is part of downstream processing. So it means that it's a process that is used to separate whatever the product of interest from the broth. Yeah. So yeah, so it's actually a downstream processing, and it is applied not just in bioprocess but also in various industry, including wastewater and other industries. Okay, another one is filtration. Filtration is by using filter, right? And when we say about filter, when we talk about filter, you should know that filter, it comes in various pore sizes. There is pore sizes. You know what pore? So you know filter, right? Let's say you imagine the filter. It is not just, it has like some kind of like holes, right? And that we don't say holes, we call it pores because it's very small. Yeah? So that pores is actually the one that it allows certain things to come through, and it retains certain things. So it has, different filters have different pore size. Yeah? So what differentiates one filter to another is the pore size. So filtration is also a process that is used to separate the toxic materials from the suspension, whatever the suspension is, right? By using filters with appropriate pore size. Okay. So if you know that, let's say, if you want to filter maybe pathogens, you should know the size of the pathogens in order to make sure that the pathogens are to be retained on the filter rather than going through. If it goes through, it means that the filtration doesn't work, isn't it? Right? So you should know the rule of thumb of this process is that you should know the size of the things that you want to filter. If let's say you want to filter, let's say whatever, let's say arsenic or maybe whatever the toxic compounds, you should know the size of that particle first, yeah, before you buy the filter, before you do the filtration. Okay, right. Filtration is a physical process. Yeah, it's a physical process because it doesn't actually alter the chemical composition of the materials. Yeah. The problem with the filtration is that it's not mentioned here, it's considered as quite an expensive process, yeah, because of the cost of the filter, yeah, expensive. And also another problem is the clogging problem. Yeah? So once the filtration takes place, right, over the time, when the things are accumulated on the filter, it will affect the efficiency of the process. We covered this in bioprocess as well. So that is the basic attributes of filtration. Okay. Right. So those are the physical processes: filtration, centrifugation, sedimentation, and flotation. So which can be applied in order to separate or to remove the toxic materials. So the third non-biotech method that we are going to look at is thermal processes or incineration. Incineration means like burning the materials, but we don't call burning, we call incineration. So basically, the toxic materials, they are burned, right, in a certain or in a special device or equipment, so that the toxic waste are no longer there. And this is quite common for hospitals or for laboratories that want to get rid of pathogenic waste materials, yeah, so that the pathogens are no longer there. Yeah? So incineration is one of the processes that is used to remove the toxic materials. Right. We study about production of biogas, right, in LU5. So one of the benefits of incineration, although it might cause some pollution, but the benefit is it can actually generate heat and power. It can generate electricity or energy, or what we call biogas. If let's say the starting materials is organic compounds and organic materials. So that is one of the benefits of, right? So here, this is examples of the equipments or facilities for incineration. Yeah. So here is just to introduce you several types of incinerators. Incinerators are those equipments that are used for incineration processes. Okay. This is liquid injection. Okay, I just go through briefly. Okay. Rotary kiln. Yeah, you can see this one is in vertical orientation, right? This one. Right, you can see the temperature is very high. It's not 100 degrees, but more than that, way more than that. It's 1000 degrees Celsius. Huh? So you can imagine that the heat is really massive. Huh? And also we study about, oh yeah, it's not that, it's not incineration, that is. Okay, so this is incineration. So this is another type of incinerator, rotary kiln, for it, yeah, which is a matter of different design or maybe different mechanisms, yeah. Right, this one is fluid bed. Okay. So this is the schematic diagram of it. But the main principle is to burn whatever the toxic materials and generate the heat at the same time. Okay, advantages of using incinerators. So having high temperature processes, like just now you see that it's more than 1000 degrees Celsius. So it's actually an excellent way of killing the toxic materials, especially if it is related to pathogens like viruses or whatever the pathogenic bacteria. So those pathogens can be burned or can be killed effectively by using thermal, by using incineration. So that's why it is an excellent disposal technology for, especially for those kind of materials or toxic materials. It offers detoxification of all combustible carcinogens and mutagens, yeah, which may otherwise result in disease transmission. Okay. And then reduction of leachable waste from landfills. Okay, so instead of like disposing the materials at the landfills, if you opt for incineration, it can avoid the problems of leachable waste. Is those waste that leak into the ground. For example, if you dispose whatever the toxic materials at the landfill, at the end, those toxic will go through the ground and it will eventually contaminate the groundwater, right? So those are the leachable waste. So instead of having that kind of situation, if you incinerate it, you can actually avoid the soil pollution. And yeah, and one of the significant advantage is the energy recovery. Yeah, so incineration, it produces energy. Yeah, whereby the energy you can actually use it for many applications, for running, for having, for the electricity, for the heat. Yeah, especially in cold countries, they can use the heat to for the heating equipments, for the heating system of the buildings or whatever the area, yeah, provide the heat. Okay. The disadvantages of using incinerators, the capital investment is high. So it's not that every place can practice it, yeah, because the cost is expensive. And also it requires highly trained operators. Yeah? So we must have expertise, certain expertise to conduct the process. Yeah? Right, so it comes with the expertise, with the cost as well. Okay, the second part is the hazardous site clean up. Okay, hazardous waste, toxic waste is not that really much different. It's something that poses substantial potential threat to public health. Okay? I just go through here. Hazardous waste are materials that are known, or tested to exhibit one of the four hazardous traits: ignitability, reactivity, corrosivity, or toxicity. Okay, so those are the traits that define the waste as hazardous. Okay, so this is the degree of ignitability. Okay, you can see the rating. This is a standard rating, from 0 to 4. From 0, you can see the water is the least hazardous one, right? And if you see the rating 4, those are the flammable gases, the propane, butane. Okay, so they are actually a standard that categorizes the degree of those things. Actually, it's not just ignitability, but also the corrosivity, reactivity. But I don't have it here. Okay. So I just want to highlight the 9 stages. The 9 stages to a site clean up. So whenever you whenever a site, a place is contaminated, and we want, I mean, certain authorities, they want to clean up the site. So these are actually the stages that they have to go through. Okay. So first they identify the site, right? And then they do preliminary investigation, right? Perhaps to identify what are the toxic materials present and what are the environment, yeah. And then what is the short-term cleanup action? So you can see actually it's not a very straightforward process. It involves like several stages before finally the place can be cleaned up from the toxic or hazardous materials. Short-term cleanup actions, you can see here, short-term, and then remedial investigation or feasibility study. So this one is like is actually a small-scale studies, small-scale studies conducted in order to see whether the approach that whatever the approach chosen is suitable or not. Yeah, because the reason why is because the party might not want to incur the cost of loss. Let's say the approach is not suitable to be applied for the whole site. Okay, if let's say they don't have these steps, the short term and this remedial investigation, they might incur big loss, big cost of loss. Yeah, if let's say the whole area, let's say they have invested whatever the approaches for the whole area, and it turned out that it doesn't work, and it will cause that big loss. So that's why these small scale studies is important. Okay, and then after the remedial investigation, there is a remedial action plan, action design, and then this is actually the evaluation process. Okay, so they are evaluating whether the approach at the small scale work or not. Okay, and before, at the end, they go for the operation. This is the operation at the large scale. And once they operate the process, of course, they have to maintain it. They have to maintain from time to time. So that's how it comes at the end, the maintenance. Okay, so just to show you here, it starts with several stages of steps before the large scale operation of the cleanup is conducted at the site. Yeah? It's not that really much on that. Okay, and then the last part is bioremediation. Okay, this is the core part of the topic, yeah, because it involves the biotech approaches. Right, so apart from all the three methods that I discussed, that I mentioned just now, the chemical, the physical, and also the thermal, right? Bioremediation is another way how we can treat the toxic waste, the hazardous waste. right? And of course, this is our concern because we are biotechnologists. Yeah? So bioremediation is a process, is a process that is that uses microorganisms to transform harmful substances to non-toxic or less harmful compounds. Yeah? So this is the name of the process. So just now, when I introduced you the works done by researchers, bioremediation of mercury, bioremediation of cyanide, bioremediation of what else? Lead, right? So bioremediation is a process. Whatever the things that is degraded, it can be specific. It depends on whatever, the toxic materials. Yeah. But bioremediation is a generic process. Yeah? It's a generic process. Yeah. So by definition, it's like it uses microbes to degrade, to transform or degrade. Yeah. Actually, these harmful substances, they are actually food to those microbes. Yeah, food. They eat the food. Those harmful substances, they are harmful to us, but they are not harmful to the microbes. That's why they eat it, So that is the science behind it. So they eat those food, those substances as food, right? And metabolize. When they eat, they produce, they metabolize and produce non-toxic compounds. So at the end, if let's say the lead, whatever the toxic materials, the mercury, once they are exposed to these microorganisms that can eat them, at the end, these toxic materials will no longer be there. The mercury will no longer be there. The lead will no longer be there. So that's how these toxic materials can be discarded. We can get rid of these toxic materials. Okay. Right. So you can see here, bioremediation uses naturally occurring microorganisms such as bacteria or fungi to degrade the harmful chemicals. So I introduced just now the term indigenous, right? Indigenous. Well, you should know by right now what indigenous means. Indigenous means like something that is natural, yeah? Or we also call like wild type, but we don't say wild type in this. We normally wild type when we say want to compare with the recombinant. But you should know that natural, wild type, indigenous, they are referring to the same meaning. Okay. Yeah, so this is correct, but then it can also trigger this knowledge about the indigenous microbes. It can also trigger improvement studies, yeah, through the genetic engineering, right, and also recombinant DNA technology or synthetic biology. Synthetic biology is another field of biotech. So this will lead to recombinant microbes, right, that can be used to perhaps enhance the natural process of bioremediation. Okay, so I hope that you get the concept clear. Okay, for example, when soil bacteria are exposed to organic contaminants, strains of bacteria often naturally appear that can break down these substances to obtain energy. I think I've covered the points here. Yeah, microbes degrade organic waste, including synthetic organic chemicals, such as right. So the environmental factors that can slow down or affect the biodegradation process. So this is basically related to naturally occurring bioremediation. When I say naturally occurring bioremediation, it's like the bioremediation that happens as it is in the environment. It is not under the control of humans. Okay, like it happens. If let's say you don't have a look at it, it happens there. Okay, so what's the problem with this naturally occurring bioremediation? Sometimes even though the microbes are there, but sometimes the concentration of the chemicals could be too high, which is not perhaps which are let me rephrase, which are maybe too much for the microbes, for the naturally occurring microbes. So some of them might be left untreated. Some of the contaminants might not be eaten by the microbes because there are not much microbes. So that is one of the problems. So what happened is that it will slow down. Perhaps it happened, but maybe it takes longer time than the optimal time. Okay. Another one is the number of type of microbes may be inadequate. Yeah, they are microbes, maybe they are naturally occurring microbes, but maybe they are not enough. That's one thing. And another thing is they might not be as efficient as we hope for. So that is what to be solved by recombinant DNA technology, where we want to have maybe a more productive kind of microbes that can do much better degradation. Okay, so that is examples of the problems with the natural bioremediation. Okay, the conditions may be too acidic or alkaline. So the conditions here is referring to the environment, to the environment of the environment of the that surrounds the materials, and also the microbes. So when the condition is not optimal, what happened, even though there are microbes, there are food over there, but it might not be favorable for them. Yeah, this one is pH, kan? This is related to pH, and we know that every microbe, it has its own optimal pH. Yeah, some microbes might not work efficiently under acidic pH or under basic pH, so they might not degrade the things effectively. Okay, so that's one of the problems. And then lack of specific nutrients. Okay, because when it is like happen naturally, no one cares. So there are some nutrients that are not enough, that are not there for the microbes. Maybe the microbes need certain additional nutrients like nitrogen, phosphorus, for them to grow better. But because of the nutrients are not there, so they might inhibit the growth. They might inhibit the metabolism of the waste, the toxic waste. Okay, it's same like human. Like if let's say we don't have enough vitamin, get certain vitamins, we may be still alive, but we might be maybe weak, or we might not be as efficient as those that have enough nutrients. The same goes to the microorganisms or organisms. Right. Moisture conditions may be unfavorable. This is still related to the condition. Same. Yeah. So moisture means like the water content. Yeah. Maybe it's too dry or too wet. Yeah. So if it's too dry, means that it's not enough. Like us, if we don't have enough water, we might not we might be weak. So the same goes to the microbes. The conditions, the water conditions or the moisture condition is also important for the microbes. And that can affect, in this case, the biodegradation. And then a lack of oxygen, nitrate, sulfate. Yeah, so these are all the nutrients. Same points with the one before. Nutrients, oxygen, because some of the microbes, they are aerobic, highly aerobic. Some of them may be facultative aerobic. So for those highly aerobic, they need a lot of oxygen. So if let's say they don't have enough oxygen, they might not be effective in their metabolism. Yeah? So these are all the environmental factors that can affect the naturally occurring bioremediation. Okay, naturally occurring bioremediation. Yeah, I think I've mentioned about degradation of these cyanide or these some of the compounds, toxic materials. in the slides before. I think this is quite an old information. Asbestos there is a research done. So actually, some of the information, let's say, like in 2021, so some of the compounds might not get, some of the compounds might not get, how to say that? Macam ni lah, like this one. This is actually from the years before. But as the years goes by, like new discovery has been found, right? And some researchers, they have found that the bacteria can degrade, for example, bitumen. So perhaps in 5 years or 10 years time, you can see that the information is increasing. Yeah, the knowledge about the biodegradation will be better than in the previous years. So like this one is asbestos. I think there is also there is already a research done on the asbestos. Okay, so these are examples, some more examples of research on bioremediation. So this one is on fungal leachases. So actually, when we talk about the microorganisms, what actually drives the bioremediation or biodegradation process is the enzymes that they secrete. Okay, so those enzymes that are actually work on the metabolism, that work on the degradation of the things. So leachases is one of the enzymes secreted or released by fungi or yeast, and the enzymes is important for the bioremediation. Yeah. So one of the examples of the application is for the remediation of colors. Remediation of colored waste from textile industry. And just now we talk about the chemical treatment, kan? Like how chemical treatment can solve the problem of colored compounds from textile industry. So by adding certain types of chemicals. But apart from that chemical treatment, if we want to use the biotech approach, we can apply certain types of enzymes or organisms, and one of them is leachases. Yeah. So here I just want to give an exposure for you, like what people have done on the research, yeah, related to bioremediation. Okay, so this is another example of the roles of leachases. Yeah, degradation of chloroamphenicol. Yeah, and the leachases is from this fungi, yeah? This Tramatus hisutal. So there are different fungi that can produce leachases actually. Okay, advantages of bioremediation. So what are advantages of bioremediation? Perhaps we can relate with the processes that we discussed before, the physical, the chemical, and also the thermal processes. Yeah? For example, the chemicals. Chemicals involve the use of chemicals, right? Chemical treatment involves the use of chemicals. It means that they are not environmentally friendly. They might result in emission of side products that are hazardous. So instead of treating whatever the toxic materials, the process also emit the hazardous byproducts. So you can see that the cycle is not is never ending. Yeah. So another way of solving or avoiding such problems is by using biotech approaches. So bioremediation, it can eliminate, yeah, the emission of those chemicals that are otherwise produced from the chemical processes or chemical treatment. Okay, so that one is this point. And then the first point, the existing microorganism can increase in number when the contaminant is present, when the contaminant, when the contaminant that is their main food is degraded. So it means that actually, because of the microbes, whenever they eat, they grow, right? And they can actually increase in number, right? So the process can happen actually naturally, and that is one of the advantages. So the amount of the microbes can increase by itself, whenever the food or the contaminant are there. Okay, the third point here is less expensive than other technologies. If you want to compare with the chemical treatment or the thermal treatment, incineration, we know that it is an expensive process. So instead of using those treatment, we can opt for bioremediation, which is less expensive. Yeah. Another advantage is that bioremediation can be accomplished on site, meaning to say that you can do the processes at that particular site of contaminated area, yeah, or we call it in situ. If you want to use chemical processes, perhaps you have to take the contaminated samples and you have to treat it somewhere else, right? So it's not on site. Yeah? So bioremediation can be conducted on site. Yeah? So what is the benefits of the on-site treatment? It eliminates the need to transport large quantities of contaminated waste off-site, and also it can eliminate the threat to the human, right, during the transfer of the contaminated waste, yeah, during the transportation. So the reason why it can be accomplished on site is because we can actually just like let the microbial degradation to happen at that site. Yeah. So instead of removing the things. The disadvantages of bioremediation, the most significant one is in terms of the time, yeah, because it's a biological process. It takes time. Yeah? It doesn't happen overnight. Yeah? So that is one of the most significant disadvantage. It takes longer time compared to chemical treatment. Chemical processes, normally it doesn't take that really long time, as a longer time than biological processes, yeah? Right. And also research is needed to develop and engineer bioremediation technologies. This is true, because it takes time to actually for us to understand the metabolism of this bacteria in degrading whatever the toxic materials like cyanide or whatever lead. It takes time for us to understand, and from there that will trigger another research. Okay, let's say we identify the bacteria that can degrade mercury, for example, and from there it will trigger perhaps creation of recombinant microorganisms. So it takes time actually. It's not that it will not take 1 or 2 years. It actually takes like more than 5 years or even more than 10 years to understand entirely. And even that we don't understand yet, yeah? Because the knowledge about microorganisms is very massive, I can say. And then in some cases, depending on the parent compound, byproducts may be formed. Okay? I mentioned just now that chemical process it may produce hazardous compounds, right? It doesn't mean that the bioremediation does not produce byproducts. It may also produce byproducts, but perhaps the level of toxicity, yeah, some of them might be toxic, but some of them, most of them might not be as toxic as those emitted by the chemical processes. Okay, so in some cases where the byproducts are toxic, that is a disadvantage of bioremediation, same as the chemical processes. Limited to biodegradable compounds only, others may need chemical treatments. Okay, so this one is subjected to studies as well. Okay? Some of the things might not yet be studied, maybe, yeah, or might not be able to degrade by the microbes. So in that case, chemical treatments is still needed. The methods in soil bioremediation. Okay, so now we have a look at bioremediation for soil. Okay, so bioremediation, it doesn't only occur on the ground. Yeah, it can also happen, or it can also be conducted for the polluted water in the sea or in the river. Yeah? So here it's just an example. So don't restrict your understanding to just saying that bioremediation is only for soil. Okay, so this is just an example. So there are two types of bioremediation. Yeah, they are in situ and ex situ. In situ means situ lah, dekat situ. In situ, on site. On site. Okay. And another one is ex situ. Ex situ means it's not at the site. The contaminated samples, they are taken to different places and they are treated at different places. It's not at the contaminated site. Okay, so that's the difference. So for in situ bioremediation, what kind of approaches can be done? Okay, in situ, you just imagine that we are treating the contaminated areas. Yeah. So one of the approaches is biostimulation or bioventing. So what is biostimulation and bioventing? Is the addition of certain nutrients to the place, yeah, in order to trigger or in order to enhance the biodegradation process at that contaminated site. So just now, when we talked about the limitation of, some of the environmental problems, right, for the naturally occurring bioremediation is the lack of nutrients, the lack of certain nutrients needed by the microbes to do the biodegradation. So in that case, that is why biostimulation or bioventing approach is created. That is to provide the nutrients to the contaminated site in order to trigger the naturally occurring microbes at that site to degrade the toxic materials. Okay, so this picture shows you how the biostimulation or bioventing is carried out. Yeah, it involves like a pump, yeah, pumps, whereby the pump, the nutrients are pumped through into the soil. Yeah, you can see here is the injection wells, the injection wells. Okay, so what is inside the pump is the nutrients. Normally is nitrogen, phosphorus, nitrogen and phosphorus, because those nutrients are needed for the growth of the microbes. It can increase the growth rate of the microbes. So nitrogen and phosphorus are pumped into the soil. That is biostimulation. So as the name implies, is to stimulate the growth, biostimulation. Okay, apart from nitrogen and phosphorus, another type of nutrient or compound that is injected is hydrogen peroxide. Hydrogen peroxide. Okay, why hydrogen peroxide? Is because it provides, it is actually a liquid oxygen. So it will provide oxygen to the microbes inside the soil. So this is an example of the pump that pumps the hydrogen peroxide, yeah, to stimulate the activity of the naturally occurring microbes in the soil, so that the microbes can do the degradation better compared to if, let's say, the oxygen is not provided. Okay, so this is an example of the pictures that shows the pump, yeah, that is built in order to pump the hydrogen peroxide to the soil underground. Yeah? So I don't go through word by word. I just want to highlight the points, important points. Right, that is in situ bioremediation. Ex situ bioremediation. Ex situ means it is not at that place. Means that the contaminated samples, they are taken to another place where the process or the treatment is carried out. Yeah, that is ex situ bioremediation. Okay, so where the contaminated stuff is taken, it is taken to the bioreactor. Okay, I know that you have come across the word bioreactor before, right? In bioprocess. In bioprocess, we talk about bioreactor is a vessel, but actually bioreactor in broad definition, it doesn't only refer to confined vessel. Bioreactor, it can be any place or any platform where the biological degradation happens. Even your stomach is also bioreactor. Your stomach, yeah? Your guts. So this is, in this case, the bioreactor is a place or is a site where it contains the contaminated samples, and the treatment takes place inside that, the biological degradation. Okay, so don't limit the term bioreactor to only the vessel. Okay. So you can see here, this is a slurry bioreactor. This is where this is actually the contaminated materials or samples, yeah, that are taken to this slurry bioreactor, right? And this is where the process is controlled, is maintained, yeah, takes place. And here is where everything like the oxygen is provided, the nutrients are aided, and the mixings are performed. Yeah? So the advantage of this method, ex situ bioremediation, is that it is easy to control the process because it is a specific place, yeah, like slurry bioreactor is a specific place to carry out the bioremediation. So everything is aided easily, like the nitrogen, I mean the nutrients, the mixing, the oxygen, yeah. So and also the observation, the control, the pH. I mean everything is at that site, yeah. So it's actually much easier compared to the in situ bioremediation. Right. So, but the choice, how people choose whether it is ex situ, whether it is in situ, it depends on the case lah. Some of the cases, maybe the toxic materials are not suitable to be transported. So they have to be treated in situ. So some of the things, maybe it doesn't involve that really much, maybe, I don't know, maybe it doesn't involve that massive amount, or it's not that really, I mean, it's possible to be carried out, then we go for ex situ. Okay, so the choice depends on the case. So that's all about the in situ and ex situ bioremediation. Okay, final slide is about final slides are about just a case. Yeah, it's just what happened in 2010. I just want you guys to relate with the real incident happened. So in 2010, there was an incident called Deepwater Horizon oil spill, or Gulf of Mexico oil spill, okay, which happened in the Gulf of Mexico. Okay, so it's actually basically it's an oil spill lah. But what makes this event very popular is that it is the largest marine oil spill in history. Yeah, so meaning to say that it covers like very big area of the sea. Yeah? So if you see, this is the map of USA. This is USA. It's not the full map, but this is actually USA, and this is Mexico. So the incident happened here. This is what we call Gulf of Mexico. So that is where the incident happened. So what happened is that the oil is spilled to the sea. So you can imagine that a large amount of oil is spilled, and the effect is very massive. It caused, you see the picture over here. This is where the oil looks like, yeah, on the sea, the oil spill. And also it doesn't actually just affect the sea, but also the beach. Yeah? You can see the beach over here. Right. So to say that the effect is very massive, the effect is very massive. And this is one example of a real case scenario that perhaps people have to think about whether how to apply maybe bioremediation. I don't know what happened in that year, but this is an example of real case scenario that requires bioremediation or requires the treatment of the toxic materials. Okay, so what I would like to emphasize here is the microbes. Because okay, let me go through the slides first. So following that incident happened in 2010, the oil spill. So people, after that incident, people have, the researchers, they try to, to see or to study about about the microbial community surrounding the contaminated area. Yeah. So these are some of the researches done. Yeah. For example, here, the first paper here is published in 2013. They tried to evaluate the bacterial community structure in oil collected from the sea surface and sediment in the northern Gulf of Mexico. Okay, so you see the term the sea surface and sediment, because whenever the spill happens, it just first it will go through the ground of the sea, right? And that's how that's how actually how the oil contaminated the sea. It's not just the surface, but also towards the ground in the sea. Yeah. So this group of researchers, they try to evaluate the community of bacteria, whether they are bacteria or not, that comes or approach that area, yeah, whenever those oil present. Yeah, so you can see from their studies over here, they found out that there are high abundance of Methylococcus. I just read here. Methylobacter, Entinobacteria, whatever lah. Banyak lah bacteria ni. Okay, so those are bacteria that they found out that reside the oil area. Yeah. Okay. So meaning to say that why these bacteria are there? Because these bacteria can actually eat the oil. They have the potential to eat the oil, basically. So that's why they are there, because the oil are food to them. Yeah. Yeah. So another study is this one: succession of hydrocarbon degrading bacteria in the aftermath of the Deepwater Horizon oil spill in the Gulf of Mexico. So this is another study. There are actually many studies. If you just type Gulf of Mexico oil spill, you can see lots of studies that comes after the incident, and they try to understand the bacteria or the microorganisms that reside in that particular area actually. But I will not go through in detail. I just want to introduce you, those studies. So this is another study: hydrocarbon degrading bacteria and bacterial community response. So you can see the term here: hydrocarbon degrading bacteria. Hydrocarbon is referring to the oil. So why those bacteria are there? Because they are the oil. The oil is the food to them, and there are actually many types. It's not only one type of bacteria or microbes. Okay. The last one, I want to share this one just now, actually. Any questions so far? Do you understand what I'm discussing? Okay, just want to show you what's the microbes that eat the Gulf oil spill. Yeah, so these are that I found on the internet. Okay, thanks for the response. Okay, you can see these are some of the findings about the types of microorganisms that the researchers have found. Yeah, and these microbes actually they eat the oil at that site. Right? So this one is the name is quite not that really common, is it? Thalassolitis olivorans. It's a bacteria. Is it a bacteria? It's a bacteria. No, it's one type. Can you see the screen? Can you see the yeah? See it? The picture that I show you, yeah. Okay, this is another bacteria. Oh, I don't see here. Wait. You can see the picture of the microscopic picture of the bacteria.
No, doctor.
No. Wait. I think now will be possible. Yeah, this one. Okay, this one. This is some of the pictures. Yeah, the bacteria that they found at the Gulf. Ni apa ni? Gulf Mexico. Tu. So yeah, just to show you, this is what is this? Leptonomonas, the genus, the name. When they found the things, when they isolate whatever the bacteria, they have to characterize it. You know, they have to find the identity of it. Yeah. So that's what they have identified lah. This one, okay, this one, and then this one is what another alkane eater. What is this? Olispera. The genus is Olispera. Yeah? When they characterize it, they found out this is fall under the genus called Olispera. This is Oceanospirillus. I think that that's it. So the name is very rare lah, because they are not. I mean, they are not present in common environment. They are present in those areas that are that have the oil. Yeah, Colvella genus. Yeah? This is the oil-eating microbes can be found. So whenever the oil present at the Gulf Mexico, yeah, the spill happen. So these microbes, they will go towards that area actually. So that's what happened basically. That's how they can isolate those microbes from that area. This one is Alkaniborax bocumensis. Has played a role in oil spill cleanup. So the good thing about this bacteria, they helps actually in the natural bioremediation. They helps to eat, they actually eat the oil, and when the oil is eaten by them, it means that the oil is clean from the area. Yeah. So that's the roles of these microorganisms. Okay, I think that's it. All right. So that's it for this topic. Yeah. So basically, what we have discussed here is so what we have discussed in this chapter or in this LU is the toxic waste treatments. Yeah, what is toxic waste? And first, we have a look at the non-biotech methods: chemical, physical, and thermal. Right, and then we go briefly into what are the stages or steps that needs to be taken into consideration when we want to clean a hazardous site, just the 9 steps or so. And then the last part is the bioremediation. What is bioremediation? And also some potentials, limitations, the methods in soil bioremediation, in situ, ex situ. And also I introduce you guys to the real case scenario, the oil spill at the Gulf Mexico. Yeah? All right. So any question? Anything not clear? We want to